So what happened? In a very short period of time, we had these bright blue cells, and when we interrogated them, there wasn't a single molecule of any kind from the original species in the cell. So when we sequenced the proteins, every protein molecule came from the new chromosome that was put into the cell. So it became very clear that DNA is a life software system. And if you change the software, you change the species. So we had done this now with native chromosomes, so we thought we had everything we needed to go ahead and do this with the synthetic DNA. We had to find a way, though, to get the bacterial chromosome out of the eukaryotic cell yeast to transplant it back into bacteria. And it turns out we had to methylate the DNA to get all this to work. But we developed this circle where we could just take any bacterial chromosome and grow it in yeast as a eukaryotic chromosome. And the whole difference is just adding this synthetic yeast centromere. So maybe the difference between prokaryotes and eukaryotes is just this simple change in the genetic code. We can make any changes easily in that bacterial chromosome using the yeast genetic systems. We can isolate it, transplant it, and create a new species. So we thought we solved everything. We decided to go ahead with the new chemistry now and make the much larger genome of the M. mycoides, which is 1.1 million letters of genetic code. And to do this, we used Dan's new system. He actually made it quite quickly. We started with 1,000 base pair pieces. We put 10 of those together to make 10,000 base pair pieces. We put 10 of those together to make 100,000 base pair pieces. We put 11 of those into yeast, and yeast assembled the genome. We did the transplantation, and it didn't work. So we had to develop what software engineers have of debugging software to find out where the error was, because we had designed many parts of this genome, and we needed to find out what was wrong with it. And to make a long story short, we found an error, a single letter deletion in one gene in one of the 11 100,000 base pair pieces. We corrected that error, redid the assembly, did the transplantation, and we got the first ever synthetic cell. So it just shows you how important the genetic code is in having it accurate. In this case, one letter out of 1.1 million made all the difference between life and no life. It can make the difference in traits, it can make the difference or no difference at all if it's in a region that doesn't matter. This was in an essential gene, and knocking out that one letter caused a frame shift and nothing worked. So initially we only got one cell, but on repeating it, we got a number of these and had a nice press conference and a big paper in Science and a lot of excitement. But I learned starting back here with Nate Kaplan that extraordinary claims need extraordinary evidence to back them up, and that's a constant problem in science. And so one of the things we worried about from the beginning is that even one molecule of native genome, if it came across as a contaminant, it could fool us into thinking we had created synthetic life. So we created a unique way of watermarking the genome so that we could prove unconditionally that it was our synthetic DNA, and also would mark the species in the future as clearly being a synthetic. And these watermarks, we actually invented a new code to write the entire English language with numbers and punctuation using the 4 letters of genetic code. We're not the first ones to do this, but people had done it wrong in the past. They basically used an ASCII code, and that's what George Church recently published with trying to encode a book in DNA. And the problem with that is it's not biologically viable. We created a code that puts in very frequent stop codons, because the last thing you want to do is put in a quote from James Joyce and have it turn into a new toxin that kills the cell. Maybe you can only get quotes from bad literature and have it do that. But so by putting in frequent stop codons, we will never add a new biology to the cell. The first watermark contained how to decode all this new information, and also being the first species to have a computer as a parent, it had to have a URL built into its genetic code. So as people solved this code, they would send an email to the species proving that they had solved it, and eventually then we put the whole thing on the internet. So the rest of the watermarks contain 46 names of all the scientists that contributed to the research, the names of the institutions, and the names, and 3 quotations from the literature that I added in, because with our first genome, we didn't put anything from the literature, and people were complaining that we didn't put in like one small step for bacteria. So we tried to step up the game a little bit. I don't know if people can read the quotes. The first is from James Joyce: "To live, to err, to fall, to triumph, to recreate life out of life." I thought that seemed appropriate. The second is from American Prometheus, Oppenheimer's biography: "See things not as they are, but as they might be." And the last from Richard Feynman: "What I cannot build, I cannot understand." And after this became public, obviously there was a lot of excitement about this notion. And then we got a letter from James Joyce's state attorney complaining that we hadn't asked permission for the quotation. James Joyce was dead. We didn't quite know how to ask him. But it turns out in the US you can use up to a paragraph as long as you attribute it properly. So that sort of went away. And then we started getting an email from a Caltech scientist saying we'd misquoted Richard Feynman. But if you look on the internet, this is the quotation that you find. And we argued back with this guy, and finally he sent a picture of Feynman's blackboard at Caltech with his famous quotation, which is actually a much better quotation. It turns out his biographer inverted it somehow. So it's, "What I cannot create, I do not understand," which is, I think very pertinent to understanding the genetic code and being able to create life from it. So we've gone back and corrected the errors in the DNA so that Feynman can rest peacefully. So what is the next stage with all this? Well, we're doing an experiment right now where we've actually designed the first genomes ever completely from scratch in the computer. And we're building those now to see, in fact, if we can have a definition of a minimal genome and if it will give a living cell. And we've been discovering a lot of genes around the planet that I like to view as design components for the future, the same way the electronics industry in the 40s and 50s started with resistors and capacitors and ultimately transistors and integrated circuits. At Synthetic Genomics, we actually have computer software for designing new life software and doing this for a variety of very specific purposes. And I won't talk any more about this tonight other than that we've had some real success now designing and modifying, improving the efficiency of photosynthesis so we can actually get much greater efficiency with sunlight, with some algae that we're creating down the road. But people have been talking, Larry mentioned a little bit in the introduction, about how the future is changing very rapidly. So the future of manufacturing is going to be all distributed manufacturing with things like 3D printing. And the key issues will be either the instruments, the IP around it, or the cost of raw materials. And so you can build in a 3D machine these very, very complex devices. And these are now cheap enough. Many people have these at home. I've ordered one. But instead of making inanimate objects, we'd much rather go in a more exciting direction of trying to print DNA. Here's an example, and obviously there's a very good local company doing this, trying to print skin cells, but we're back at the basic DNA level. And if you think of DNA as digital information, it turns out we can send it at the speed of light. And so we're calling these biological teleporters, and there's a lot of different implications for that. If you're living on Mars, which nobody is yet, but within 2 decades, as SpaceX or others have their way, there will be colonies on Mars. And so you can get biological material to or from Mars simply as digital information, as an electromagnetic wave. So the government's been trying to find out how to reduce the cost from billions and billions of dollars to get samples back from Mars. Our notion is, and I'm certain that we will find life, it's a matter of digging deep enough into the crust on Mars, of sequencing the DNA there and just sending it back, and then recreating it in a spacesuit lab here, because everybody's worried about the Andromeda strain and sample return from space wiping out the Earth. So at least we can do it completely in a secure lab and just rebuild the species here. There's more practical and immediate implications of this, in this with pandemics. So we've been working on the influenza virus for some time. My institute is a major sequencing hub and an emergency response unit for the US government in Washington, where we're tracking the genome of flu from birds, from pigs, from humans around the world. But many of you will remember with H1N1, it took about 9 months to get a new vaccine out. 250,000 young people died in the US. Everybody thinks it wasn't a bad pandemic. It wasn't as pandemics go, but a whole new group of young people were obviously very adversely affected by it because they had no prior immunity. And just to remind people about the 1918 pandemic, about a third of the world's population was killed from the flu virus. Today, a third of the population would be 210 million people. And if you saw the recent movie Contagion, I think that's not a great scenario to go through, and I think we can prevent that. Right now, flu vaccine is manufactured in the US in about 900 million chicken eggs. That creates about 300 million doses of vaccine. It's a 50-year-old at least technology, and we found out we could change this entire process. So we've been working with BARDA and the US government